Initially, there was some question, why work on bone? Why are you in the Dental Institute? Well, my answer is you can't have teeth without bone. Of course, the craniofacial complex is shaped by bone, and that's part of our identity. The major questions that we have been trying to answer really have related to determining the nature of the bone marrow stromal cells, skeletal stem cells, what makes them self-renew, that's one of the primary characteristics, and also what guides the fate that they choose to take. They can make bone, they can make cartilage, they support blood formation, and they make marrow fat. I've worked extensively on developing techniques for bone regeneration using bone marrow stromal cells. We developed this transplantation system where we take the cells and we're transplanting them into immunocompromised mice and rats. And if it's a genetic disease, those mutant cells will recreate abnormal bone that is virtually identical with the bone that comes from the patient with the disease. And this was actually one of our major assays that we used in the early studies of fibrous dysplasia of bone and the McCune-Albright syndrome. So we can use these skeletal stem cells as models of disease. Other uses have been high throughput screening for identification of drugs that may be useful in treatment of osteoporosis and also other types of bone diseases. It's my hope that I can get a clinical protocol off the ground someday for regeneration, maybe of jawbone. But more recently, we've had some pretty interesting results in cartilage formation.
We're trying to look at how cartilage develops and the entire synovial joint so that maybe we can find ways to help regenerate it or engineer tissues from stem cells to try and recreate the tissue. Synovial joints are found in our knees and our jaws. They help us chew. And when the cartilage on the ends of the bones degenerates, it can become very painful, and that cartilage really never repairs on its own. And this can lead to osteoarthritis and pain. We've actually developed our own custom device for culturing cartilage micropellets, and that consists of an array of little microwells that has a little mesh over it. And the mesh is small enough for single cells to come through, but once they come through, they aggregate into a sphere, and the sphere is too large to come back through, so it stays within this little compartment.
In about 10 days, you can see that pellet has grown and it's white and glistening, and it's made a very, very nice cartilage. We've found a scaffold that seems to support the formation of really stable cartilage, and this has been a problem that has faced the field for a long time.
If we're able to successfully make stable cartilage, we can then transplant these into people's cartilage defects and hopefully prevent the further degradation of cartilage and osteoarthritis, and of course maybe avoid a hip transplant.